All right. So now let's discuss, now that we kind of have an overall idea of what a GWAS is and kind of what it gives us as far as experimental output, let's talk about the statistical concepts that underlie this particular analysis. And kind of what makes these GWAS go, in a sense, is this statistical concept called linkage disequilibrium. And what I really want us to understand, in a very basic sense, is that it's a measure of the tendency of two genetic markers to be inherited together on the same chromosomal segment. And so let's think, let's have a phenotype of interest, some certain trait. So let's say that way back in evolutionary time, we have a particular genetic mutation that causes, let's say, purple hair, for example. And so at the time that this mutation arises, this entire chromosome is in perfect linkage disequilibrium with this causative mutation. And so it's inherited along with that particular mutation. And as we move forward in evolutionary time, right, we have recombination events occurring in meiosis, and in the next generation, we have a particular portion of our chromosome that's been largely recombined, and so this black portion here is no longer in linkage disequilibrium with our original mutation. And as we move forward through the successive generations, we have sort of a narrowing down of this this window, sort of how I like to think about it, this window of linkage disequilibrium where this genetic material is still inherited along with that original mutation. And so, for example, if we perform genotyping and we genotype a particular SNP that is within that window of linkage disequilibrium, we will see a statistical relationship between this particular genotyped SNP and the mutation that we're trying to identify. And so it all goes down to, we're trying to identify the causative mutation. And so, for example, if we genotyped a particular SNP in this black region here that's largely recombined and doesn't have any relationship to the original mutation, we won't see any statistical relationship. And these windows of LD form what are called haplotypes or haplotype blocks, which is defined as a set of DNA variations that tend to be inherited together. And SNPs in a haplotype that differentiate observable forms are called tag SNPs. These tag SNPs are used in the creation of these DNA microarray chips that we use in these genome-wide association studies. And so these microarray chips, as we sort of talked about in our introduction, can simultaneously interrogate hundreds of thousands to millions of these tag SNPs across the genome at an extremely reasonable cost. And this method is an extremely high throughput method, so we can do thousands of samples very, very quickly, measuring the single nucleotide variants in those samples. And so, for example, we see here our genetic material, and we see an A to C SNP here. And so when we start measuring it in a number of individuals, right, we have one individual that's homozygous for the A allele, one individual who's homozygous for the C allele, and one individual who is heterozygous. And so we do this. So here's SNP1. We do this for hundreds of thousands to potentially millions of SNPs across our genome. And these are manufactured and produced by biotech companies. Some of the leading brands are Affymetrix and Illumina. Illumina, remember, produces a lot of the next generation sequencing machines that we get a lot of our data from. But basically, what we're looking for is the increased prevalence of a particular version of one of those tag SNPs in a large population of cases versus controls. And so this tells us that the causative mutation may have been inherited along with that particular tag SNP. Remember, because of that concept of linkage disequilibrium, right? Like kind of tagging along and being inherited along with that particular single nucleotide variant. Now, a common representation of GWAS data that we'll be looking at is known as a Manhattan plot. And they call this a Manhattan plot because it sort of looks like the Manhattan skyline, right, with these skyscrapers of genetic association, essentially. So if we see increased prevalence of a particular version of one of those tag SNPs, we see kind of this, what's called an association peak, or one of these skyscrapers here. So what we're measuring here, we have chromosomal position on our x-axis. So remember, humans have 22 autosomes. And so these are our 22 chromosomes here. And what we're plotting it against is our p-value. And a p-value is simply the probability that the difference we see between our cases and control population is by chance alone. And so taking the log base 10 of that p-value, basically it takes our smallest p-value and it kind of flips it on its head. And so it makes it, it gives it a higher value here. And so our lowest p-values we see towards the top of our plot here, making these association peaks. So the higher this peak, the stronger our statistical relationship is between our genotype, the particular version of that SNP, and our phenotype of interest. And so whatever that may be, whether it be obesity or type 2 diabetes or asthma or whatever we're studying. And so here's kind of a closer look at that Manhattan plot. And so all of these colored dots that you see here are indicative of those single nucleotide polymorphisms that were genotyped on that DNA microarray. And so all of these are telling us that these SNPs are statistically associated with our phenotype of interest. And so these sort of act as signposts to kind of go look in that particular region of the genome more in depth to see if we can identify, what genes are actually there and what might be, having an impact on our phenotype of interest. And so remember, to our discussion about this idea of genetic architecture and complex traits, right? So our rare alleles, we use whole exome sequencing, whole genome sequencing, and our more common alleles, we use the DNA microarray to perform a genome-wide association study. And you can see that we've, identified common variants with small effects by GWAS, and so we've performed GWAS in pretty much any disease phenotype that you could think of. Here's just a few examples. So LMTK2 in prostate cancer, gets identified with GWAS. And so in a lot of the complex disorders that we might study, we often find, several genes or potentially hundreds of genes that could be associated with a given complex phenotype. And so here's kind of a rundown of asthma loci or genetic variants that have been detected regarding the asthma phenotype by GWAS. And so you see a number of them associated with different genes kind of across our genome. And here's just one example. So remember, what we're looking for is this indirect association because of that linkage disequilibrium. So I like to think of this as kind of like guilt by association, right? And so all of these tag SNPs here, right, are inherited along with this causative mutation within this particular gene. And so this is an example of chromosome 17, a very well-known asthma locus that remains one of the most well-studied and replicated loci. You can see it here with a nice association peak in our Manhattan plot telling us that the statistical relationship between genotype and phenotype is extremely strong. And all of these genes, you see there's a number at this particular locus on chromosome 17, that's this well-known asthma locus. They fall into three main categories based on the function of these genes, right? They function in the immune system, they function in mucosal biology and function, and lung function, which makes a lot of sense when we think about the overall asthma phenotype, right? It's a disease that has a lot to do with not only the proper functioning of our immune system, but also mucosal biology and the functioning of our lungs. But remember, because this is a complex trait, each of these genes, or each of these variants that we find associated with asthma in this case, makes a very small contribution to the overall disease. And after genes have been found to be associated with a given phenotype, we now have to figure out kind of how they work in asthma, right? We need to piece that together still. So there's still further validation to do with these particular studies. And of course, the work is always ongoing, especially with these complex phenotypes.